Welcome to session 19. In this session we are going to discuss about the factors affecting enzyme action, coenzymes and inhibitors. The objectives of today's class are understand the factors that affect the enzymatic activity, define cofactor, understand the types of enzyme inhibitors. The outcomes are knowing the factors influencing the enzyme activity, learning the meaning of cofactors, to be able to list out the types of inhibitors. Now, in our previous classes, we have seen that enzymes are biological catalysts which accelerate the rate of chemical reaction. The enzymes are proteins which has a substrate binding site called as active site. Okay, and we have also seen that the attachment of the substrate induces conformational change, so induced fit model by Koshland, and the binding of enzymes to the active site can be inhibited by something called as inhibitors. So these are the things which we have seen in our previous sessions. We have also seen the properties of enzymes, like they are highly specific, they are sensitive to temperature, also sensitive to pH, and sensitive to concentration of the substrate, and they are stereoscopic as well as inhibited by inhibitors. Stereoscopic refers to they are stereospecific actually, refers to a condition where a particular enzyme is highly specific for a stereoisomer. So stereoisomers, you have heard about isomers. So the L and D forms, or plus or minus isomers, that means the certain molecules rotate the plane of plane polarized light in the clockwise direction, and some rotate in the anti-clockwise direction. So accordingly you have levo rotatory and a dextro rotatory molecules. So a stereoisomer is a levo rotatory or a dextro rotatory isomers, and these enzymes are highly specific to a particular type. So certain enzymes are specific to L types and some are specific to D types. Okay, so that's what stereo specificity is. They're stereospecific in nature. These are the properties which we have discussed in our previous classes. So in today's class we are going to see about the factors that affect the enzyme activity. So if you are thorough with the properties of the enzymes, then this understanding this is much easier, or at least you are going to get the explanations of those properties. Now the first factor that influences the enzyme action is the temperature. And we all know that enzymes are proteins, so that means they are formed of amino acids. The temperature sensitivity of enzyme is because of its proteinaceous nature. Since proteins are sensitive to the temperature, enzymes are also sensitive to temperature. Now how they are sensitive? It's because these proteins are formed of amino acids, and these amino acids are not just present in the form of primary structure, that is simply in the sequences. Instead, they show different levels of folding. Okay, and these levels of folding are maintained at a specific temperature. As soon as there is a change in variation in the temperature, these foldings are altered, and that's how it affects the performance of the protein, or in other words, the performance of the enzyme. Okay, and here most of the enzymes are highly specific, and the activity of the enzyme usually increases for every 10-degree increase in the temperature, which is called as a Q10 effect. Okay, so now in case of most of the enzymes, the Q10 is 2 between 0 to 40 degrees Celsius. That means the reaction rate increases by 2-fold for a Q10 of 0 to 40 degree Celsius. That means for most of the enzymes, the activity is almost 0 at 0 degree Celsius, as you can see here on the graph here. Okay, so this is the graph what can be used to understand the activity of the temperature, effect of temperature on enzyme activity. So it is almost close to 0, or maybe very less. So as you increase as you keep on increasing the temperature, what happens? Those active sites of the enzymes become much active. Okay, so the protein folding will be conformational changes will help in opening up the active sites. So now what happens? Because of that there is a increase. This is the rate reaction, and this is the temperature. X-axis refers to the temperature. So as you keep on increasing the temperature, so rate of reaction also increases. It increases and almost reaches optimum highest at 40 degrees Celsius. At 40 degrees Celsius. So that means by this time almost all the enzymes are active, and the active sites and the binding of enzyme substrate complexes takes place. But what happens as you increase the temperature, further increase in temperature, so above 50-60 degrees Celsius, denaturation of the protein takes place. Now I can explain it by using the example of egg. You have all seen that the white portion of the egg is almost translucent, the albumin part in an unboiled egg. But as soon as you boil it, what happens? It becomes semi-solid and solid, it solidifies. And this translucent to solid nature, liquid to solid nature of protein is because of coagulation. Okay, and that is the effect of temperature on protein. Similarly, in case of enzymes, as the temperature increases beyond 40, 50, so 50, 60 and above that, usually after 60, denaturation of the protein takes place. That means the protein folding is altered. So once the protein folding is altered, that particular protein will not be able to perform its normal function. That is what the enzyme is inactive, or it is denatured above 60 degrees Celsius. But it is not true for all of them, because there are certain enzymes which are active even at a higher temperatures, like Turk polymerase, Thermus aquaticus. So from that polymerase enzyme has been isolated. This enzyme is active even at a temperature of 90 degrees Celsius. Also venom phosphokinases, the phosphokinases enzymes that are present in the poison, so they are active above 100 at a temperature of 100 degrees Celsius. Even muscle adenyl kinase, these are the enzymes which are active even at a higher temperature. So there are also certain enzymes which are active at a higher temperature, and there are proteins which are active at a very low temperature. So those are exceptions, but most of the biologically active enzymes are highly active at a temperature of 40 degrees Celsius. So, and above 50 to 60 degrees Celsius, the denaturation happens. Okay, and most of the enzymes are inactive above 60 degrees Celsius. So that's why you are going to see this graph when you plot the activity of the enzyme against the temperature. Okay, and this can also be applied in case of, that's the reason why actually we store the food in freezers. So because most of the enzymes are inactive, the bacterial enzymes are inactive at a lower temperature like 4 degrees Celsius to 8 degrees Celsius. So what happens? Enzyme activity doesn't take place, so that means food doesn't get spoiled. And we also know that we boil food and keep, and that is because we are going to kill the bacteria. In other words, we are actually denaturing the enzymes. That's how we preserve the food. Okay, so the temperature plays a very important role in affecting the activity of the enzyme. And remember Q10 effect. So what happens here is that for every 10 degrees Celsius in the raise in temperature, there is increase in the activity, but to a certain extent. Beyond that, this doesn't happen. The next one is pH. Okay, pH is nothing but the active ion concentration, active hydrogen ion concentration in any sample, in any solution rather, is called as pH. So the more the hydrogen ion concentration in the solution, more acidic it is. Less the hydrogen ion concentration, less acidic it is, or close to neutral it is. Okay. Now this hydrogen ion concentration plays a very important role again in enzyme activity, because again the fact is enzymes are proteins. And hydrogen ion interferes or interacts with different amino acids that are present in the protein or polypeptide chain. Thus, they are either activating the enzyme or inactivating them. Now, every enzyme has its own specific pH. So, in temperature we talked about a general one. That means most of the enzymes have optimum temperature of 40 degrees Celsius. Okay, 40 to 45 degrees Celsius, but most of them they become active about 25 degrees Celsius. So, you can take a range of 25 to 45, that's where the enzyme activity is seen. for most. That is a general one, but this is not true for the, pH is not, in case of pH it is not true, because every enzyme has its own specific pH, optimum pH, and it is not general. For example, the enzymes that most of the biological active enzymes are active at a neutral pH. A pH of 7 to 7.2 is highly optimum pH for the enzyme action. For example, salivary amylase. So it is active at neutral pH. Whereas the enzymes of your stomach like pepsin, they are active only at acidic pH. The pH of 1.2 to 2.2, highly acidic pH, is favorable for pepsin because hydrogen ions will activate these enzymes. Whereas when you take the example of trypsin, that's an enzyme active in the intestine, secreted by pancreas. This enzyme is active at a alkaline pH, neutral to alkaline pH. So 7.5 to 8 or 8.2, that's a pH range for this trypsin activity. That means there are enzymes which show, so if I have to draw a plot, so already we have one, so let me just take this one. Now if you are talking about pepsin, the activity is highest here, okay, and if you are talking about this trypsin, the activity is highest here. Whereas there are other enzymes which show activity, highest activity at neutral, near neutral pH. So this point, or in other words, these temperatures are the optimum, these pHs are the optimum pH for these enzymes for their activity. And all these are because of the interference of hydrogen ion with the active site. So highest concentration of hydrogen ion might activate a active site of enzyme like pepsin, whereas the same higher concentration of hydrogen ion will denature, okay, the active site of enzyme like trypsin, and similarly salivary amylase. Okay, so that means a lower concentration of hydrogen ion will activate the trypsin and salivary amylase. Okay, so each and every enzyme has its own optimum pH. Some of the enzymes have optimum pH in their acidic range, and some of them have in alkaline range, and most of them have in neutral range. So pH plays an important role in enzyme activity, and these somehow regulate the digestion of food in different parts of your alimentary canal. The next factor that influences the enzyme activity is the substrate concentration. And substrate, as you increase the substrate concentration, what happens? The rate of chemical reaction also increases, okay, but it reaches a peak at particular point and then it is maintained at an equilibrium. Now the graph what you are seeing here is, see, there is an increase, there is slow increase in the beginning, then there is a, this rate increases to a larger extent, then it reaches a plateau, okay, a plateau, and it is maintained at equilibrium, same, it is maintained in the same plateau. Now why this happens? Why we are getting this arc, okay, close to a parabola? Now this happens because, so when there is a, when the reaction begins, we know that substrate is going to induce the enzyme for its active site. So at this time the enzyme activity is low, because some of the enzymes are still substrates are trying to bind to the enzymes, the active sites of it. Now as soon as this binding continues, more and more enzymes are active and a large number of substrates are going to bind. So at time 0 there is no enzyme substrate complex. At a time 1, okay, 1 I am just taking as a unit, it can be a second, minute or hour. At a time 1, so let us say that 10% of the substrates have already bounded to the enzymes. At a time 2, let us say that 25 to 30% of them are bound. And at a time, okay, say n at a time n, so most of them, let us say that 50% of it is bound. And maybe at time n plus 1 or n plus 2, almost all the enzymes which are present are now bound with the substrate. Let us say that there are some 1000 molecules of substrates and there are some 500 molecules of enzymes. Now at that particular moment, so now here when I am talking about this particular this particular point, what has happened, the enzymes, 500 enzymes are there. How many substrate can bind to 500 enzymes? 500, maximum 500. Okay, so that's where you reach this point, a plateau. No further additional rate can be increased because all the enzymes that are available have already active sites are occupied by the substrates, and this 500 it continues until all the substrates are converted into products. Okay, so substrate concentration increases the rate of chemical reaction. When all the substrate enzymes are bound with the substrate, there is no further increase in the rate. That doesn't mean the enzyme activity is not taking place. There is no further increase in the rate of the chemical reaction. It reaches a equilibrium. Okay. The next one is very important, enzyme concentration. Enzyme concentration. So, increase in concentration of the enzyme will also increase the rate of chemical reaction, and it is a straight line. So, that always depend on the substrate concentration. When there is supply of n number of substrates, unlimited supply of substrates and unlimited supply of enzymes, the rate of chemical reaction keeps on increasing. It keeps on increasing because enzyme concentration is unlimited. But the hindrance point is, as soon as the substrate concentration is stopped or further supply of substrate is stopped, the enzyme activity stops. Okay, so enzyme concentration also plays an important role in enzyme activity. So, temperature, it is the temperature, it is the pH, it is the enzyme substrate concentration, it is the enzyme concentration. Not only that, there are also other factors that influence the enzyme activity, like cofactors, coenzymes and the prosthetic group. Presence and absence of inhibitors also influence the enzyme activity. So, cofactors, let us see how cofactors are influencing the enzyme activity. Before that, let us see what is actually a cofactor. Okay, a cofactor is something that is bound to with the enzymes. Now, enzyme as a whole, when you are talking, it is a protein complex with the exception of riboenzymes anyway. So, this protein complex, it may not be active by itself. It may not be active by itself. That means it might need some activators. So, these activators might be organic substances or it might be inorganic substances. So, an active enzyme is a protein part of the enzyme plus some activators that are present. Now, let us call that protein part of the enzyme as a apoenzyme, okay, and the non-protein part of the enzyme can be called as a cofactor. Now, there are some confusions about use of cofactors and coenzymes. Some of the authors use the word coenzymes for the organic substances that are part of the enzyme. Some prefer to call them as cofactors. So, in my discussion, I will use the word cofactor as the non-protein part of a complete enzyme. Okay. Now, to avoid the confusion, let me write certain points here. So, active enzyme which is called as a holoenzyme. Okay, a holoenzyme, complete, a complete enzyme, holoenzyme. Okay. This holoenzyme is equal to apoenzyme plus a cofactor. I'll just write only cofact here. So, now what is this apoenzyme? It is the protein part of the enzyme. Okay, and the non-protein part of the enzyme is called as cofactor. Most of the enzymes are inactive if they are having only cofactor. If these enzymes are bound with the if they are having only apoenzymes, if this apoenzyme is bound with the cofactor, then it becomes active, and such an active enzyme is called as holoenzyme. Okay, an enzyme is active when it is in the holoenzyme form. Holoenzyme is formed of apoenzyme and a cofactor. What is the apoenzyme? The protein part of it. What is the cofactor? The non-protein part of the holoenzyme. Okay. So, these cofactors are smaller molecules. They help in the improving the catalytic property of the enzymes. Now, there are two types of cofactors. They are coenzymes and the prosthetic groups. Again, there are different explanations for these cofactors. Now, a cofactor can be an organic one as well as it can be an inorganic one. Okay, there can be an organic cofactor, there can be an inorganic cofactor. In organic cofactors, there are again two types. They are coenzymes and the prosthetic group. So, when we are talking about two types of cofactor, here I am referring to the organic compounds that are forming the cofactors. I will repeat to avoid the confusion. Holoenzyme is formed of apoenzyme and a cofactor. A cofactor can be an organic substance or an inorganic substance. Under organic substances, it can be a loosely bound organic substance or it can be a tightly bound organic substance. If the organic substance is tightly bound, then it is called as a prosthetic group, and if it is loosely bound, it is called as a co-substrate or a coenzyme. Okay, it is called as a coenzyme or co-substrate. So, let me write it here. So, what is a cofactor? A cofactor is either organic or it is inorganic. Okay. So, in case of organic, there are two types. One is coenzyme, the other one is prosthetic. Okay, prosthetic group. So, here we are talking about these two types. First one, let us say a coenzyme. A coenzyme. Now, what is a coenzyme? A coenzyme is an organic molecule. Organic molecules are familiar. They are the compounds containing carbon, hydrogen and oxygen, isn't it? So, they are organic compounds. How they are different from prosthetic groups? These organic compounds which are loosely bound with an enzyme, okay, which are loosely bound with an apoenzyme are called as coenzymes, and they have low molecular weight and they can be separated, okay, by simple dialysis process. Coenzymes can be separated by simple dialysis process. They are loosely bound, okay, and they are organic molecules of lower molecular weight, and they act as group transfer reagents. And the examples for such coenzymes are ATPs, NADP plus and NAD plus and NADP plus. There are also coenzymes which are derived from vitamins. Vitamin derived coenzymes are present, like thiamine pyrophosphate is derived from thiamine, okay, and it transfers aldehyde or group keto group, okay, and the enzyme which is dependent on them is a transketolase. And there is FMN, riboflavin, so hydrogen and electron, they are L-amino acid oxidases are dependent on them. So, like this, there are a number of such coenzymes derived from vitamins. So, then there are nucleotide derived coenzymes like ATP, NAD plus and NADP plus. NAD plus and NADP plus. These are coenzymes. Okay, they are loosely bound to their enzymes and they are of low molecular weight and they are organic molecules. Okay. Now, these coenzymes are also called as co-substrates because they will bind with the enzyme just like a substrate binds. But a substrate is converted into product, a co-substrate is not converted into any product. Okay. So, next one. Yeah, here we have different examples also like coenzyme A, coenzyme Q10, then the water soluble vitamins B complexes, okay, vitamins NADH, NAD plus I said, no. So, these are all examples for coenzymes, loosely bound organic molecules. Then in addition to that, we also have prosthetic groups. Now, these prosthetic groups are tightly bound with the enzyme. They are the integral part of the enzymes and they cannot be separated by simple dialysis processes. For example, FMN and FAD. Okay, in certain cases, FMN and FAD, they are going to act as a prosthetic groups. They are tightly bound with the enzyme and they are the part of the enzyme complex, multi-enzyme complexes as a whole. In addition to this, there are also inorganic cofactors. These inorganic cofactors include something like these metal ions. Okay, they can be called as metal activators. Like chloride ion is an example for ion activator that activates enzyme amylase. Okay, there are also enzymes like ATPases. They are activated by magnesium and calcium ions, metal activated ones. So then there are metalloenzymes. For example, alcohol dehydrogenase and aldolases have zinc in them. So these are the ions, inorganic ions. They are going to act like a cofactor. Okay, they are going to act like a cofactor. That means if an enzyme have to be active, they need them. In the absence of these metal ions or anions, the enzyme will not be active. Okay, so that's about these coenzymes, coenzymes and the prosthetic groups, as well as the metal activators. I can also give the example of copper, that is metal ion, which is very important cofactor in case of cytochrome oxidases. Zinc we have already seen. So these are going to activate the enzymes. In their absence, the enzyme is inactive. So repeating, cofactors are some molecules that are needed for the activation of the enzymes. They can be organic or inorganic. If they are organic, they can be coenzymes or co-substrate as well as prosthetic groups. Then inorganic, they are simply the ions, activator ions. They can be metal ions or some anions like chloride. Okay. Then we are moving to the next concept, that is the enzyme inhibitors. So enzyme activity is also influenced by inhibitors. As the name itself says, they are going to inhibit. They are going to suppress the activity of the enzymes. They disturb the functions of the enzymes, and because their presence will influence the catalytic activity of the enzymes. Sometimes they will hinder the binding of the substrate to the enzyme, or in some other cases they will not alter the binding process, but they will influence the catalytic property of the enzymes. Now these inhibitors may be metabolites or non-metabolites also, and there are two types of inhibition. One is reversible inhibition, the other one is irreversible inhibition. First let us see irreversible inhibition. As the name itself says, the binding of these substances to the active sites or to the enzyme will affect the activity of the enzyme irreversibly. That cannot be changed. That means the total enzyme inactivity takes place. Irreversible. The binding and the inhibition cannot be reversed back by any process. That is called as irreversible inhibition. A classic example for it is diisopropyl fluorophosphate, which is also known as a DFP. Okay, diisopropyl fluorophosphate, DFP. This is actually a nerve gas, and it was actually developed by Germans during the Second World War. Okay, and this can bind with the serine residue. Serine is an amino acid of the enzyme, particularly enzymes like acetylcholine esterases, which are involved in muscle activity. Now what happens when this binds with the serine residue? They are going to bind to the active sites of the enzyme. The acetylcholine esterase enzyme, it will be inactivated. This will result in paralysis. Okay, paralysis of the body. So that's how this gas was used by the Germans during the World War. Interesting, isn't it? So similarly, there are also enzymes like malathion. These are the pesticides which are organochloride pesticides. They also are irreversible inhibitors, and they bind with the acetylcholine esterase, and they will inhibit the contractions of the muscle, thus resulting in paralysis. This malathion can also be a inhibitor in case of humans. Okay. And then there is iodoacetate. This activates on, this binds on glyceraldehyde 3-phosphate dehydrogenase. It binds with the active site. Since binding takes place with the active site, no substrate binding occurs, permanent damage of the enzyme takes place. So no activity. This particular enzyme is active enzyme in glycolysis. So you won't find the conversion. So product formation doesn't take place. Okay, so that's a irreversible inhibition. Then there are inhibitions which are reversible. That means with the certain, by changing certain conditions, the inhibition can be prevented. There can be two types. One is competitive inhibitor, the other one is non-competitive inhibitor. A competitive inhibitor, as the name itself says, they compete with the substrate. Okay, they compete with the substrate for the active sites. So once a competitive inhibitor binds, substrate cannot bind with the active site. So that is a competitive inhibition. Now for example, succinate dehydrogenase is an enzyme and whose substrate is a succinic acid. And there are other analogues, structural analogues we call it. That means structurally they are similar to succinic acid, like malanoic acid, then glutaric acid and oxalic acid. These are analogues. They are structural. They show structural similarity with the succinic acid, and these they bind with the active site of the enzyme succinate dehydrogenase, and that means succinic acid cannot bind with the succinate dehydrogenase. Now all of them are competing for the active sites. Now what is the solution? Most of the times, by increasing the concentration of the succinic acid, this can be reversed. So as the concentration of the succinic acid increases, affinity of the active site towards the substrate increases, towards succinic acid increases. So now there is a difference in the concentration of the competitor and the actual substrate. So they get detached from the active site, the succinic acid can bind. So the inhibition can be reversed. Then there are also like alcohol dehydrogenase enzyme, one more dehydrogenase enzyme, and its substrate is actually methanol. This methanol is converted into formaldehyde by alcohol dehydrogenase. It's a normal reaction. But ethanol is a structural analog to methanol which can compete with the methanol. You know, there are cases of alcohol poisoning. Where methanol is taken by mistake, as by accidental conditions. Now what happens? This methanol will be converted into formaldehyde, which is highly poisonous one, highly toxic to the tissue. So this can be treated by using ethanol. Methanol poisoning can be treated using ethanol. Very interesting. So ethanol competes with the methanol and it binds with the alcohol dehydrogenase. It is converted into acetaldehyde. Acetaldehyde is less dangerous. So that means the treatment, it is a temporary treatment anyway. So, but anyway, useful. Now here we are using the reversible inhibition nature of the enzyme, but actually it is used in the opposite condition. Okay, inhibition can be it has many medical clinical significances, and it is used in different kinds of treatment too. And one of the classic example, as I just told, using ethanol as a competitor for methanol in methanol poisoning. Okay, so that is a competitive inhibition where two compounds compete for an active site. Next one is a non-competitive inhibition, and here they bind to enzyme at a site different from the active site. Okay, so see here, I can just draw a diagram. We have discussed about it in our previous class too. So here is the active site. The inhibitor will bind to next site. Okay, next site. It won't bind to the active site. It will bind to a site other than active site, and that means substrate can still bind. Substrate can still bind with the active site, but enzymatic activity doesn't take place because this particular inhibitor will affect the catalytic activity. You know, in active site there are two regions. One is binding site and the catalytic site. So binding site is not inactivated. Instead, catalytic site will be inactivated. So what happens? Enzyme activity doesn't take place. In certain cases, the active binding site itself is influenced, denatured. Then no further binding of substrate 2 can see. That means non-competitive inhibitor either will inhibit the activity by altering the active site, binding site, okay, of the active site, or by altering the catalytic site of the active site. By any means it is going to influence. Again, this is a reversible one by different means, by changing the concentrations of different ions, by changing the pH, by changing the temperature, the inhibition can be reversed. Okay. So, that is all about the factors affecting the influencing the enzyme activity: temperature, pH, okay, substrate concentration, enzyme concentration, cofactors and inhibitors influence the enzyme activity to a larger extent. Okay. Let us see how much you have understood. Increase in temperature for more than optimum level results in increase rate of reaction, decrease the rate of reaction. The rate of reaction is not affected, denatures the activity of enzyme. Increase of temperature more than optimum level, and the answer is denatures the activity of the enzyme. Factors that influence the rate of reaction are temperature, concentration of hydrogen ion, concentration of substrate, all of the above. Yeah, answer is all of the above. All three of them have influence. Okay, next one. All three of them have the influence. Salivary amylase requires dash pH for normal functioning. The enzyme present in saliva 4.2 to 5 to 6 or 6.2 to 7.2, 1.8 to 3.2, 7.6 to 8.6 saliva. Okay. So, it is 6.2 to 7, near neutral pH is needed. The substance that enhances the activity of enzymes by binding it is said to be the substance that enhances the activity of the enzymes: coenzyme, inhibitor, cofactor, competitor. The answer is cofactor. Even though there are certain confusions about coenzymes, cofactors are the broader categories of those molecules. In non-competitive inhibition, inhibitors bind to. In non-competitive inhibition, inhibitors bind to other than active site, inhibitory site, enzyme substrate complex. Other than active site. So, it can bind to an enzyme itself, or it can also bind to an enzyme substrate complex too. Okay. So, that is all for today's class. Thank you.